All right, welcome to Lecture 2, and we're going to talk about the molecular basis of hypertrophy. What pathways underlie hypertrophy. So there's been a number of pathways identified as particularly important for muscle anabolism, and those include the mammalian target of rapamycin, or mTOR, Mitogen-Activated protein kinase, and various calcium-dependent pathways. One thing is these pathways, they do overlap at key regulatory steps, and there's evidence that a lot of these pathways act synergistically. They're not just redundant, but they act synergistically. All right, so this is sort of what a pathway looks like. As you can see, it's extremely complex, but almost too complex to say what are we affecting with training. Well, one thing I sort of want to zoom in on here is mTOR, and again, it's mammalian target of rapamycin, and this is a key player in hypertrophy. Why? Well, we think that mTOR is a perfect sensor for cell growth. So there's mTOR right there. It senses amino acids. It can sense mechanical stimuli like lifting weights. It can sense growth factors like IGF-1 and insulin, energy status of the cell as well. So how's it regulated? Well, there's a number of ways. One thing is if you look at mTOR, it acts on the ribosome to initiate translation. So we call that translation initiation. So you're basically getting the process started off. If you look upstream of mTOR, you can see AKT. And basically that you have a pathway called the PI3K, AKT, mTOR signaling pathway. And PI3K is activated by growth factors like IGF-1 or insulin. When that's activated, it inhibits a complex known as TSC1 and 2, or tuberous sclerosis factor 1 and 2. Basically, this complex, TSC1 and 2, inhibits a protein called Rheb. Right? And Rheb, when it binds to mTOR, increases its activity. Basically, mTOR is a protein kinase. When it's activated, it phosphorylates downstream targets, and that turns on protein synthesis, both acutely and enhances long-term impact of protein synthesis. MAPK, which is known as the cell's fuel gauge, basically triggers the binding of TSC1 and 2, which can inhibit Rheb. So we have this interaction, say, between growth factors via the PI3K AKT signaling pathway as well as MAPK. But the key is, again, you want Rheb to bind to mTOR, and lots of things are working on that. Classic study by Bodine in 2001. What you're seeing, the CH, is essentially the normal training stimuli in this rat model, and this is an increase in size from at day 7 after day 14 after extreme hypertrophy bouts, and you can see the CH is increasing in muscle size, but the CH plus RAP is not. Well, RAP equals rapamycin, which inhibits mTOR. That's why mTOR is called mammalian target of rapamycin. Well, rapamycin inhibits mTOR. So when you inhibit mTOR, you don't get hypertrophy. So this tells us that this is a critical regulator of skeletal muscle growth. If we were to make this a little bit more complex here, you go again to the mTOR pathway. What you see is IGF-1 and insulin. Also MGF, which is a mechanically induced form of IGF-1. You can see it's induced by things like muscle damage. Will bind to the IGF-1 receptor or insulin receptor, activate AKT, or PI3K, which activates AKT. That inhibits TSC1 and 2, allowing Rheb to bind to mTOR and stimulating translation initiation. However, you can also, if you're training super high volume, or if you're doing cardio, for example, you're going to run out of ATP. That's going to increase AMP, which means the cell's low in energy. AMPK, or AMP kinase, is going to sense that your cell's low in energy, and it's going to increase the formation of TSC1 and 2 inhibit Reb and now decrease translation initiation. Amino acids are thought to partly work by increasing the association of Reb with mTOR by a number of complex mechanisms. We may also have hormonal interaction. Hormones tend to increase the actual transcription. So, for example, testosterone acts directly on the nucleus and increases the transcription of myofibrillar proteins. And that's just sort of an overall interactive interplay between mechanisms of hypertrophy on mTOR. Now, IGF-1 or insulin-like growth factor 1, we have three different versions of this, IGF-1EA, IGF-1EB, and IGF-1EC. EC is also known as MGF or mechanogrowth factor. Like the name suggests, this is sensitive to mechanical stimuli. What we know in this study, Baumann looked at non-responders, which got zero hypertrophy, moderate responders, about 30% hypertrophy, and extreme responders, which is like 60% hypertrophy. And the extreme responders had much greater MGF mRNA, as well as IGF-1. So this says that it's possible that these are regulating differences in non-responders and extreme responders. When I was in grad school, in fact, everything centered around IGF-1 in resistance training. And I was actually in grad school after this study. But Hornberger, when I was Hornberger cast doubt on this a few years before, but it took a long time to really get into popular media. But he found that IGF-1 antagonists did not prevent protein synthesis. Now, what is this? It doesn't indicate that IGF-1 is not important. It indicates that there are other mechanisms involved. What are those mechanisms? Well, if you had to design a sensor for muscle mechanical stimuli and transduce that information to a chemical signal via protein synthesis, where would that sensor lie? Obviously, in the muscle. So if we look at a muscle here and zoom in, you know that muscles are made up of contractile units called sarcomeres, and those are connected via Z-lines. The Z-line has a great potential to sense stress and is where most of the damage occurs. So if we look here, if you look at a Z-line intact, a Z-line contains an enzyme known as phospholipase D. This enzyme produces something called phosphatidic acid, and phosphatidic acid switches mTOR on. This is how we believe that muscle senses mechanical stimuli. So what happens is, in part B, when the Z-line is disrupted, we think PLD, or phospholipase D, is dislodged into the cytoplasm, and then that triggers the production of phosphatidic acid. And phosphatidic acid serves as a switch to turn mTOR on. So this is support for what I'm saying. If you look at the cytosol and the membrane and the cytoskeleton, notice that PLD is very high in concentration in the cytoskeleton, particularly in the Z-line. So in summary, the Z-line is a perfect location to sense mechanical stress. PLD, or phospholipase D, is located at the Z-line and is dislodged during resistance training. PLD increases the formation of PA in the cell, and PA turns on the switch for mTOR and thus protein synthesis. One of the things to understand is that the stimulation of PA is contraction specific. So what you're seeing here in this model by O'Neill out of Hornberger's lab is where the arrows are pointing at 0, 15, and 60 minutes. You see the phosphorylation of P70, which is a downstream target of mTOR, is increased. However, the control group is not. Again, P70 is a downstream target of mTOR, a very important target. Now, if you look here, again, the control, that's the control, concentric versus eccentric. So concentrics not doing a good job triggering mTOR. But if we look here at the control versus stimulated here, At 0, 15, and 60 minutes, we see that phosphatidic acid, or PA, over to the graph of the right is increased drastically in the eccentric condition, but not the concentric condition. And when you do eccentric contraction, protein synthesis is higher than concentric. This is likely mediated by an increase in phosphatidic acid by the PLD enzymatically derived mechanism. So what is phosphatidic acid? It's a very simple phospholipid. It consists of a glycerol backbone with 2 fatty acids and a phosphate group. And basically, we always think of amino acids as building blocks, but also think about a cell has a cell membrane, and has a phospholipid bilayer. So a critical need for cell growth is the presence of raw materials for increasing the mass of the cell. Basically, phosphatidic acid serves as a signal for lipid bioavailability. And that's what we're seeing here. The phosphatidic acid serves, hey, we got plenty of lipid to grow, let's turn on protein synthesis. Now, on the other hand of the spectrum, we have protein breakdown. Protein breakdown essentially is targeted by a number of pathways. The calcium calpain is one of them. We have non-specific lysosomal, but where I'm going to key in on is the ubiquitin proteasome pathway. Basically, what we see here is that when proteins are damaged or unfolded, they're tagged by multiple ubiquitin molecules. And once tagged, they can be recognized by proteasomes in the cell, in this case the 26S proteasome, and then degraded. So that's very, very important to understand. This is what it looks like. You look at the ubiquitin pathway, there's 3 key enzymes that are always keyed in on, rate-limiting enzymes. You have E1, which activates ubiquitin molecule, and it actually requires energy. And then you have E2 and 3, which guide the ubiquitin molecule to the protein to be destroyed. That protein is polyubiquitinated, or several ubiquitin molecules are added to it, and then it can be recognized by the 26S proteasome complex. So that's very, very important to understand. Again, you have 3 enzymes. E1 activates ubiquitin, E2 and E3 guide it to the damaged protein to be destroyed. You have polyubiquitination, or multiple ubiquitin molecules bound to the protein, and then that can be sensed by the 26S proteasome for degradation. Here's an overview of what might happen with training. So if we look at mTOR, mTOR can be activated by PA, right? And that's sort of important to understand. We see IGF-1 can bind to its receptor, activate PI3K and AKT, which inhibits TSE1 and 2. We also know that MAPK can increase the formation of TSE1 and 2, say if you're doing cardio. One of the things I want to point out here is that resistance training in general can lead to a number of pathways being activated. And that's sort of the point of this. So if you're resistance trained, or if you have a chronic inflammation like when you're elderly, you might upregulate the ubiquitin pathway, and that might increase protein breakdown. So, we kind of want to think about all these things that are happening. One of the last topics I want to talk about is called the Mitogen-Activated protein kinase pathway. One of the key molecular guys in our lab, Ned Erk, is interested in this pathway. And so MAPK family of proteins, basically they're comprised of distinct signaling molecules in skeletal muscle. These include extracellular regulated kinases, 1 and 2. They include P38 MAPK and C-Jun NH2 terminal kinases, or junk. So we call these ERK1 and 2, P38 MAPK, or mitogen activated protein kinase, and junk. This family of proteins is called the mitogen-activated protein kinases. So, one thing I want to point out is that ERK1 and P38 seem to be regulated by volume of exercise. As such, they seem to regulate metabolic adaptations. So if you do low intensity, high volume, unaccustomed exercise, you'll activate ERK1, which you see on the left. If you do that type of exercise, you may also activate P38 MAPK. But unless you do high intensity exercise, you're likely not going to activate junk. Now, again, MAPK and ERK1 and 2 definitely seem to be increasing things like fatty acid uptake, fatty acid oxidation, glucose transport, and mitochondrial biogenesis. So you're more metabolically equipped for a training bout, such as a hypertrophy training bout. Junk, on the other hand, activates mitogenic protocols, and mitogenic simply means, for example, the activation of satellite cells become very important. But don't get it twisted. ERK1 and 2, for example, and P38 may also have anabolic signaling pathways besides metabolic. So what I want to point out here is what we're looking at is junk activation, and what you see is that average peak tension over time, that you see the greater the tension, the greater the junk activation. So that's extremely important, and it points out that you're going to have to lift heavy to activate this junk pathway, and junk's going to be critical for activation of satellite cells, for example. So let's compare some different models. This is a good friend of mine at a Vascular University, Juha Holmi. And Juha basically compared 15 reps by 1 RM, or 15 sets by 1 RM, versus 5 by 10 RM. So the 5 by 10 RM has more volume, like 7,000 versus 2,000 kilograms. And what you're looking at is P70 phosphorylation. Again, P70 is downstream of mTOR. And what you can see is that the black is 15 by 1, the white is 15 by 10. The white has greater P70 phosphorylation on serine 421 and threonine 424. However, if you look at threonine 389 on your right, which is B, you see that basically the 15 by 1 RM and 15 by 10 RM both activate that signaling on P70. What this is telling us that we have different sites or different residues that we can phosphorylate, that mTOR can phosphorylate, or other proteins can phosphorylate, and that these are sensitive to both volume, in the case of P70 and serine 421, threonine 424, as well as intensity, which is phosphorylation of P70S6K on threonine 389. You don't need to memorize those numbers, but you do need to know that there are different sites that you can activate this protein. And so different types of training may cause a more complete adaptation of the muscle. Okay, so if we're looking at here, the top graph, for example, P38 MAPK signaling seems to be increased similarly with 15 by 1 and 5 by 10 on graph A. But if you look at ERK1-2 signaling, it clearly favors with a 5 by 10. Remember, ERK1 and 2 has a metabolic component. But the thing about ERK1 and 2 is that it also may, if you look to this graph here, you can sort of see that ERK1 and 2 to the mid-right inhibits TSC1 and 2. So it can also help with stimulation of mTOR. Now, if you're looking here, if you stimulate PI3K via IGF-1 or insulin, that will activate AKT, that will inhibit TSC2, 1 and 2. But ERK1 and 2 can also phosphorylate TSC1 and 2. One of the things I want to point out here is that Erk signaling and PI3K can phosphorylate TSC2 on different regions of the complex, which might mean that their impact is synergistic during training. So one of the things I want to point out is that what activates MAPK pathway is not just volume or intensity, but also oxidative stress. So what you're looking at here is basically stimulation of or phosphorylation of the MAPK pathway. If you look at the graph all the way to the left, you see a lot of phosphorylation of the pathway. If you look at where the antioxidant is administered in the middle of E, you see that it completely inhibits phosphorylation of the MAPK pathway. So it suggests that you could inhibit growth by providing antioxidants around the training session, and that acute oxidative stress may be an important variable for triggering adaptation. So right here we're looking at is oxidative stress, and the soleus, which is almost nearly all slow twitch, the red gastroc, which is 50% slow, 50% fast, and the white gastroc, which is almost all fast twitch. Notice that oxidative stress following training is highest in the white gastroc, intermediate in the 50-50, and lowest in the soleus. Again, this suggests that fast fibers have a greater oxidative stress response. And we know that these fast fibers have a greater potentiality for growth. The other thing is if you're looking at nuclei per myofibril region, type 2 fibers have a greater myonuclei potential than type 1s. And that's what we're seeing here. Type 1 fibers also have a reduced ability to increase phosphorylation of ribosomal protein S6 kinase after resistance training. And that's basically what we're looking at here. We're looking at phosphorylation of S6K. And if you look at type 1, which is to the left of the graphs A and B, compared to type 2, you see greater phosphorylation in type 2. And again, this is a downstream target of mTOR. Okay, and then if you look to the right of this graph, which is B, and you look at mTOR, they're comparing essentially the type 1s versus type 2s. Type 1's the first two in the graph here. You see not as much mTOR stimulation, but however, actually the type 1 is the gray and the type 2 is the black. So you can see that 1 hour or 2 hours after, the type 2 have greater phosphorylation of mTOR. So this indicates that it's important to trigger stimulation of type 2 fibers if you're going to optimize hypertrophy. Not that type 1 aren't important, but type 2 do have a greater potentiality.